In this video, we will highlight some of the differences in transcription in eukaryotes. Much of transcription is the same for eukaryotes as it is for prokaryotes. The main differences are that there is more regulation on this process due to cell complexity and larger genomes. Multicellular eukaryotes have the most regulation on transcription since they have multiple cell types and need to only express certain genes in those particular cell types. Additionally, eukaryotes have three different RNA polymerase enzymes that transcribe specific types of RNA. The main one we will deal with is RNA polymerase II, since it transcribes mRNAs. RNA polymerase I's transcribe most of the rRNA genes that make up ribosomes. RNA polymerase III takes care of the rest of the RNAs, including tRNAs, one type of rRNA, and microRNAs. As we talked about previously, much of the difference in transcription in eukaryotes has to do with regulating the process. In eukaryotes, our genes have a core promoter and various regulatory elements that influence that core promoter. The core promoter itself is rather short and consists of a short region, usually a Tata box, and the transcriptional start site. The key thing here is this determines the precise starting point for transcription. By itself, the core promoter can start transcription, but this is at a pretty low rate and is termed basal transcription. Regulatory elements are important for enhancing or inhibiting transcription. These regulatory elements are short DNA sequences that affect the binding of RNA polymerase. These sequences affect the binding of RNA polymerase through their attraction of various transcription factors, which are proteins. These transcription factors bind at the -50 to -100 base pair region. That is to say, 50 to 100 base pairs before the start site for transcription. The binding of these transcription factors either enhances or silences a particular gene. Now we have seen the different elements that regulate transcription. Typically, these are divided into two categories. The first being cis-acting elements, which are DNA sequences, and the second being trans-acting factors, which are those regulatory proteins that bind DNA sequences. Transcription initiation in eukaryotes is quite complex, so we will only dig into this complexity a little bit. To that end, let's look at the three types of proteins required for basal transcription, that is, base level transcription. These are RNA polymerase II, the main workhorse enzyme, a set of 5 proteins called general transcription factors, and finally, a protein complex called a mediator. The figure here shows the basic order of binding for these different elements of basal transcription. This figure, however, doesn't really cover the role of the mediator, so I will briefly say that the mediator is responsible for interacting with transcription factors that enhance or silence transcription. These would be proteins that bind mostly upstream from the transcription start site. Therefore, it is the mediator that will facilitate transcription that is either faster or slower than basal transcription. We will further explore regulation of gene expression in a later video.